Welcome to The Deep Dive, the show where we sift through groundbreaking research, fascinating articles, and our own notes to pull out the most important nuggets of knowledge for you.
Today, we're plunging into a question that sounds like it's pulled directly from, well, a science fiction novel, yet is very much at the forefront of real science.
Exactly. Can a computer truly be programmed to simulate a brain?
It's an incredibly ambitious goal, isn't it? And to even begin to approach an answer, you really need to tackle two colossal challenges.
Okay, what are they?
First, you need an incredibly detailed, complete map of a brain's connections, its entire wiring diagram, what scientists call its connectome. You need the blueprint.
With the blueprint.
And second, once you have that static blueprint, well, that's not enough. You need a dynamic way to make that map do something, to actually predict its activity in real time.
Precisely. And our mission today is to take a deep dive into recent breakthroughs that are bringing us, well, remarkably closer to this monumental goal.
Yeah, you're going to hear about the incredible efforts underway to map not just tiny brains like a fruit fly's, but even a significant fragment of the human brain itself.
And then we'll explore how researchers are beginning to predict real-time neural activity across an entire vertebrate brain. We'll be talking about some really cutting-edge projects.
Things like the Drosophila hemibrain, the Human 801 dataset, and the Zappi bench for zebrafish.
Right.
Each one is really pushing the boundaries of what we thought was possible.
Okay, let's unpack this. Let's start with that foundational challenge, mapping the brain's intricate wiring, or connectomics, as you called it.
Right.
Because before we can even dream of simulating something as complex as a brain, we absolutely need its complete blueprint. And the sheer scale, the complexity of even a tiny brain is, well, frankly, mind-boggling.
It really is. And what's truly fascinating here isn't just the concept, but the sheer monumental effort involved. Take the Drosophila hemibrain project as a prime example.
The fruit fly, Drosophila melanogaster.
Exactly. A small creature, sure, but its brain is still astoundingly complex. Researchers focused on a significant portion of its central brain. They aptly named it the hemibrain,
Okay.
and they embarked on mapping it with, well, unprecedented detail for the time.
And when you say a significant portion, we're talking about mapping approximately 25,000 individual neurons and an astounding 20 million chemical synapses, those tiny communication points between them.
Yeah, 20 million, all packed into something so small you can barely see it with the naked eye. This was, at the time, the largest dense reconstruction of its kind.
Wow.
It provided a level of detail unimaginable.
It sounds like a Herculean effort.
Oh, absolutely. To give you a sense of the scale, it took about 50 person-years of proofreading. 50 person-years, yeah. Over roughly 2 calendar years, just to extract this data from a staggering 26 teravoxels of raw images.
Teravoxel. So a voxel is like a 3D pixel, right?
Exactly. The smallest unit of information in these volumetric scans. So imagine 26 trillion of these 3D pixels, each one holding critical information about the brain's structure.
26 trillion voxels.
Yeah.
That's just an astonishing amount of data. So how on earth did they even begin to capture something so tiny, so densely, and then make sense of it all?
Well, it took technological breakthroughs.
What were they?
They employed a combination of incredibly advanced techniques. First, new sample preparation methods like hot knife, ultra-thick sectioning were crucial.
Hot knife.
Yeah, imagine a razor-sharp blade heated to precisely slice the brain into incredibly thin 20-micrometer slabs. This method allowed them to get sections thin enough for high-resolution imaging without, damaging the delicate structure.
Okay, that makes sense.
Then they used focused ion beam scanning electron microscopy, or FIBSEM.
FIBSEM, got it.
This is really cutting-edge imaging. It uses a focused beam of ions to sequentially mill away ultra-thin layers of the sample while simultaneously taking electron microscope images of each fresh surface. And this yielded incredibly detailed 8 by 8 by 8 nanometer voxel data.
Nanometer scale. That's a level of precision almost beyond our everyday comprehension.
It really is. To be working at that scale, capturing such fine detail, is truly an engineering marvel.
But okay, once they had all these billions of images, what came next? Because just looking at pictures isn't enough, right?
Right.
You need to identify and classify everything.
Exactly right. That's where advanced computational methods, particularly machine learning algorithms, became indispensable.
Ah, the AI comes in.
They were used for segmentation, which is essentially drawing boundaries around individual cells and neuronal processes to identify them, and for synapse detection, meticulously pinpointing every single one of those connection points.
Okay.
It's kind of like having a sprawling city map made of billions of dots, and the algorithms help you find every individual wire in every junction box.
That's a great analogy.
But even with AI, the sheer complexity and the need for absolute accuracy required extensive, painstaking human proofreading afterward.
So it wasn't fully automated.
Yeah. No, not at all. It was a true human-machine collaboration.
And once they had these foundational maps, the next crucial step was, what, making sense of the sheer diversity within them.
Exactly. And this led to some pretty profound insights into how these circuits are organized. They didn't just map connections, they started classifying the neurons.
How many types?
They defined over 5,000 morphology types that's based on their shape, and over 5,600 connectivity types for roughly 22,500 neurons they identified.
That raises a big question. How do you even classify cell types when you have so much data and so many similarities?
Right. It's a challenge. They use tools like NBLAST for morphology-based clustering, so grouping neurons by similar shapes.
Okay.
And a new tool called CBLAST for connectivity-based clustering, looking at shared connection patterns.
Ah, so shape and connections.
Exactly. And what's unique about CBLAST is that it's an iterative process. It allows human experts to actually guide the clustering and refine cell type definitions based on both shape and connection patterns.
So humans are still in the loop, guiding the process.
Absolutely, leading to a truly comprehensive classification.
And this detailed classification led to something particularly fascinating, didn't it? About the fly brain's packing density.
Yes. They found that its computational regions adhere surprisingly well to something called Rent's rule.
Rent's rule. I think I've heard of that in, like, computer chip design.
You have. That's exactly where it originated. Rent's rule is an empirical observation from designing human-made computing systems, specifically about how the number of connections between components scales with the number of components themselves.
Okay.
It essentially suggests that when a system is packed as tightly and efficiently as possible, its communication connections follow a specific mathematical relationship.
And the fly brain follows this.
Remarkably well. Its adherence isn't just a quirky biological detail. It suggests that the brain, through, billions of years of evolution, has stumbled upon or optimized for the same fundamental efficiency principles that govern our own supercomputers.
Wow. That's kind of mind-bending. Like convergent evolution for computation.
Pretty much. It's like a universal law of optimal communication architecture emerging independently. A really profound insight if you're trying to build an artificial brain.
So what does this all mean for our big question? Can we simulate a brain? This dense fly connectome.
Well, it allows researchers to study whole brain properties, to understand how these intricate biological circuits are actually wired up. It's a monumental step towards truly understanding them, and therefore potentially simulating them someday.
Okay, the insights from the fly connectome are profound, giving us a foundational understanding of compact, efficient biological computation. But it naturally leads us to the grander, maybe more audacious question. If mapping a fly brain required a Herculean effort, what does that mean for cracking the code of the human brain? That must be a leap of, well, galactic proportions.
It truly is. And this brings us neatly to the HO1 project, which tackles that human frontier. Researchers took a single cubic millimeter of human temporal cortex. Now, this tissue was surgically removed from a patient to access an underlying epileptic focus and was then made available for this unprecedented research.
A single cubic millimeter. That sounds incredibly small.
It does, doesn't it? Yet the scale of the resulting data is just dramatic. 1.4 petabytes.
1.4 petabytes from a cubic millimeter.
Yep. That's 1,400 terabytes of raw image data. And in that tiny cube, they found an astonishing number, around 57,000 individual cells,
Okay.
and an estimated 150 million synapses.
Whoa. 150 million connections in that tiny space.
Yeah.
That's almost unbelievable.
It gives you a sense of the density, right? The imaging challenge alone for HO1 was immense, even more so than the fly brain.
How did they manage it?
They started with a 170 micrometer thick slab, and to capture the full 3D structure, cut it into 5,019 ultra-thin sections, each just 30 nanometers thick.
5,000 slices.
Using something called an automated tape-collecting ultramicrotome, or ATUM, imagine a highly automated, ultra-precise slicer that not only cuts these incredibly thin sections, but also automatically spools them onto tape for imaging.
Automation seems key here.
Absolutely essential to handle that volume.
Oh.
Then, each of these sections was imaged with a multi-beam scanning electron microscope at an astounding resolution of 4 by 4 nanometers.
4 nanometers, even higher resolution than the fly.
Yep. And this entire imaging process alone took 326 days, almost a full year, just for the imaging.
A year of imaging for 1 cubic millimeter, and then the reconstruction. Just like the fly brain, I assume this wasn't a solo act.
Oh, not even close. It was a massive collaborative effort. They blended advanced machine learning using sophisticated algorithms like flood-filling networks for segmentation, identifying cells and processes automatically,
Right.
and automated synapse classifiers. But crucially, this was combined with a collaborative human proofreading platform, leveraging the expertise of researchers all around the world.
So crowdsourcing the proofreading, essentially.
In a highly structured, expert way, yes. Because, as the scientists explicitly stated, manual reconstruction of data at this scale is simply infeasible for a single lab. It's a real testament to the power of global scientific collaboration.
That's amazing. And what did they find? Were there surprises hidden in that petabyte of data?
There really were. Some quite unexpected discoveries emerged from this unprecedented level of detail. For instance, they found that glia, often thought of as just support cells for neurons.
Yeah, the glue cells.
Right. They actually outnumber neurons 2 to 1 in this sample, with oligodendrocytes being the most common cell type.
2 glia for every neuron.
That's right. About 32,000 glia versus maybe 16,000 neurons. This really challenges the neuron-centric view of the brain and suggests glia play a much more active, crucial role than we previously understood.
That could change a lot about how we think the brain works. Any other surprises?
Yes, they also identified new morphological subcategories of deep-layer triangular neurons in layer 6 of the cortex. These clustered in mirror-symmetrical subgroups.
So like finding entirely new, distinct variations in the fundamental building blocks.
Kind of, yeah. Which could imply different functional roles we didn't know about.
Okay, and you mentioned connections earlier.
Hmm.
Anything new there?
Perhaps the most striking finding was what they termed powerful axonal inputs.
Powerful inputs.
Yeah, they found rare but incredibly strong multisynaptic connections where a single axon, that's the output fiber of a neuron,
Right.
can form up to 50 or more synapses on one target cell.
50 synapses from one axon to one neuron. That's huge.
It is. This discovery fundamentally challenges the long-held idea that most connections in the brain are weak and diffuse, needing lots of weak inputs to sum up.
Right, the sort of gentle nudge model.
Exactly. If these superhighways of information exist, it means certain signals could have a disproportionately large impact. Maybe allowing for rapid, decisive influence, it could really revolutionize how we model things like learning and decision-making in neural networks.
Wow. So bring this back to simulation. What does H01 mean for our quest?
Well, these incredible maps provide an unprecedented, really granular view into the human brain's microarchitecture. It's essentially giving us the foundational code, that incredibly detailed wiring diagram we need to even begin thinking about how its complex circuits operate.
We're truly getting the blueprint for a piece of the human brain.
Exactly. A really detailed, tangible blueprint.
Okay, so we've talked about mapping the brain's blueprint, the connectome, foundational first step. But as you said right at the beginning, mapping is just the start.
Right, the static map.
The real dynamic challenge is to understand and predict how these intricate networks of neurons and glia actually behave in real time, how they process information, generate behavior.
Precisely. This is the next frontier, bringing that static blueprint to functional life.
Mm-hmm.
And that's where pioneering projects like ZAP-eBench come in.
ZAP-eBench. That's the zebrafish activity prediction benchmark.
That's the one.
And its goal sounds incredibly ambitious. Measuring progress in predicting cellular resolution neural activity throughout an entire vertebrate brain.
Yeah, it basically asks, okay, if we watch X seconds of observed brain activity across a whole brain, how accurately can we predict the next ravi seconds of activity?
Like a weather forecast for the brain.
That's a pretty good way to put it, yeah.
Why zebrafish, though? What makes them suitable for this?
The choice of the larval zebrafish is absolutely crucial here. Very strategic. They are unique among vertebrates because their larvae are almost completely transparent.
Transparent. You can see right through them.
Pretty much. And this transparency allows for simultaneous optical recording of all neural activity in their entire brain at single neuron resolution using something called light sheet microscopy.
All the neurons.
Yeah.
The whole brain.
The whole brain in action. This isn't just a subset or a small region. It's something virtually impossible in other vertebrates currently.
That's truly incredible, being able to see every neuron fire in a living, behaving vertebrate. And the data they've collected for ZAPI-Bench sounds equally impressive.
It is. 4D light sheet microscopy recordings from over 70,000 neurons, collected while the fish was engaged in 9 different behavioral tasks.
So you can correlate activity with behavior, too. How do they actually record the activity? It's not electrical, right?
No, it's optical. They record calcium activity using a genetically modified protein called GCAMP.
GCAMP.
Think of GCAMP as a tiny biological sensor. When a neuron fires an electrical signal, calcium levels inside it temporarily go up, and GCAMP is engineered to fluoresce to light up when that happens.
Ah, so the light flashing indicates firing.
Exactly. It's an indirect measure of neuronal firing, but it's crucial for capturing this level of activity across vast populations of cells in a living animal. This raw data then undergoes, rigorous computational steps for motion stabilization and segmentation to track individual neurons over time.
And crucially, ZAPI-Bench isn't just a data set they've released. It's a formal, open benchmark, right?
That's key. Performance of different predictive models is quantified using something called mean absolute error, or MAE. It measures the difference between the predicted neural activity and the actual observed activity.
So it creates a clear, measurable way to track progress in brain simulation, like a leaderboard.
Sort of, yeah. It allows researchers worldwide to test their models against a common standard and see how well they do.
How are the models doing so far?
Well, early results from initial computational models, things like time series models that predict future activity based on past patterns, and volumetric video models that treat brain activity like a 3D movie, they show they can indeed outperform simple baselines, which is encouraging.
Right.
But it also highlights that there's significant room for improvement, which is pretty typical for a new and challenging benchmark like this. The gap between current models and perfect prediction is still substantial.
Okay, but here's where it gets really exciting, right? This is where the two fields we've been discussing, connectomics and activity prediction, truly converge.
Exactly. This is the crucial link. The specific zebrafish brain used in these ZAPI-Bench activity recordings is also undergoing parallel synaptic level anatomical mapping using electron microscopy.
The same brain. They're getting the wiring diagram and the activity recording from the exact same individual fish.
That's the plan. And what's truly revolutionary is the vision behind it. This dual approach will enable the future integration of that incredibly detailed structural information from connectomics, the precise wiring diagram,
Right.
directly into models that aim to predict and simulate brain activity.
So you're not just guessing the connections anymore.
Exactly. This is the crucial link bringing the structural blueprint to functional life. Instead of just statistical connections or assumptions, we'll have the real biological wiring guiding the simulations. Imagine trying to predict traffic flow in a city without knowing the actual road network that's the challenge they're overcoming.
It feels like a massive step towards actual simulation.
It really does.
So as we wrap up this deep dive, let's recap our journey a bit. We've explored these extraordinary efforts to map brains at unprecedented detail,
From the dense connectome of the fruit fly's hemibrain
to the petabyte-scale fragment of the human brain's temporal cortex. Huge leaps in mapping.
And we've seen how projects like ZAPI-Bench are pushing towards predicting the dynamic activity of these incredibly complex biological computers in real time.
It's clear that we're accumulating just an enormous amount of data, developing incredibly sophisticated tools to process it.
And uncovering surprising biological insights along the way about how brains are built and how they might operate. Things like those powerful axonal inputs, the role of glia.
The initial breakthroughs we've discussed, the sheer scale of mapping, the ingenious automated methods, these unexpected biological discoveries, and now the ability to predict whole brain activity in a vertebrate, they're all critical milestones.
Definitely moving us forward.
So let's revisit our central question. Can a computer be programmed to simulate a brain? Where do we stand?
Well, I think the answer is a pretty resounding, we're getting there.
Yeah.
Yeah. We now possess the first incredibly detailed blueprints, and we're actively building and refining the tools needed to run these complex simulations. It's no longer purely the stuff of science fiction.
It's becoming a tangible scientific pursuit.
Exactly. But, and there's always a but,
Hold on.
this deep dive also highlights the immense complexity that remains. The ultimate challenge isn't just gathering data or building sophisticated models, as hard as that is.
Well, that is it then.
It's about truly understanding the underlying principles. The principles of computation, learning, information processing that make a brain, whether it's a fruit fly's or a human's, so uniquely capable. What makes a network of neurons truly intelligent? That's the deeper mystery we still need to unravel.
Precisely. It's not just about replicating, it's about understanding. And that leads to a final, maybe profound thought for you, our listener, to consider. What might actually happen when the structural map of an entire brain, like that zebrafish, can be fully integrated with its real-time activity predictions? When structure truly meets function at that scale.
What new secrets about learning, about behavior, maybe even the earliest whispers of consciousness might that unlock?
It's a truly exhilarating prospect for the future of neuroscience, isn't it?
Absolutely. A fascinating time to be watching this field.